Hi everyone, welcome to the introduction of macromolecules. Today we're going to be discussing the synthesis of these macromolecules and along with how they separate from one another. So let's start by first talking about polymers. So polymers are macromolecules. They are also a bunch of molecules or monomers chemically bonded together. So for example, this is the individual monomer, and when they come together, they create a chain or a polymer, which is essentially the macromolecule. So these are the types of macromolecules. We have carbohydrates, lipids, proteins, and nucleic acids. And these are very large molecules, which is why they're called macromolecules. And each macromolecule is made up of specific monomers. So these are the individual substances that create the macromolecules. So for carbohydrates, it's monosaccharides, lipids, it's fatty acids, proteins, it's amino acids, and for nucleic acids, it's nucleotides. So when these monomers bond, they form through specific bonds, and each macromolecule has its own. So for carbohydrates, they make a glycosidic bond or a glycosidic linkage. For lipids, it's the ester bonds. For proteins, it's peptide bonds. And for nucleic acids, it's the phosphodiester bonds. So we know that they each have their own bonds, but how exactly do these monomers attach and break to form or break the chain of polymers? So first, they use enzymes. Well, specifically, through those enzymes, they go through a dehydration reaction or hydrolysis. And this is just a note, but the process of making polymers is called polymerization. So let's start by talking about the dehydration reaction. So this is used to make polymers, so while releasing water. So when both of the monomers attach, their hydroxyl groups come together releasing H2O, so leaving the oxygen molecule behind that forms part of the bond. So let's look at a closer example. So this is a dehydration synthesis example. So we have glucose and fructose molecule and with their own hydroxyl group. So when they attach, they create a glycosidic bond because they're carbohydrates or sugars. And due to the two hydrogens, 1, 2, and 1 oxygen molecules combining, it makes a water molecule. And this leaves out that 1 oxygen molecule. So let's look at hydrolysis. So hydrolysis is when the chain is breaking down. It's monomers from each other. And with the input of the water molecule, the H2O molecule splits and restoring the hydroxyl groups to each monomer. So let's look at an example. So when sucrose gets an input of water, the water molecule splits, which also splits the glycosidic bond, creating two separate molecules. So here we have glucose and fructose from sucrose. So we have the H2O, the two hydrogen molecules, and we have this last left alone oxygen molecule, and we also have this oxygen molecule from water. So this is how the water molecule splits to go back to its hydroxyl group. So this is just a note, but the enzyme that facilitates this reaction is the sucrase. Thank you for watching. I hope you learned something new today. Have a great day.